This morning we have great pleasure to welcome Dr. Richard Ducatelle, Professor Richard Ducatelle, sorry. He graduated as a veterinarian at Ghent University in Belgium, where he also obtained his PhD in veterinary pathology. And since 1989, Richard is professor in veterinary pathology, again at Ghent University. He is author or co-author of more than 650 scientific publications, and also he has been speaking at more than 100 national and international congresses. Besides, he is the inventor on more than 10 different patents. His research interest is mainly in gastrointestinal health, with a focus on poultry, calves, and pigs. And he has also been the mentor of more than 30 PhD theses in this field of research. So this morning, Richard Ducatelle will talk about small intestinal absorption versus small intestinal infection, focusing on the so-called enterocytes, the activity of enterocytes and their absorption, but also possibility for infection, which is a subject which we like to discuss in Tonisity because we are focusing on those enterocytes. So Richard, welcome. It's up to you, and we look forward to listening to your presentation.
Thank you, Mathieu. Can you hear me?
Yes, perfectly.
Okay, so thank you very much for the kind introduction, and welcome everybody to this presentation, which is going to focus, as Mathieu said, on the small intestine, for the simple reason that small intestine is actually the place where the absorption of the nutrients takes place. So whenever anything goes wrong in the animal with respect to the small intestine, it has direct effects on the performance. By the way, the picture shows the veterinary faculty at Ghent University campus. I think there is a ever-growing interest in this field of intestinal health and intestinal absorption of nutrients, especially for the reason that all over the world, antibiotics, so-called growth-promoting antibiotics, are taken out of the feed. So this is for the reason that antibiotic resistance has become a major issue all over the world. It has been calculated that for instance, in the US, that antibiotic resistance is costing society billions of dollars a year. So that is the reason why people are becoming very, very careful with the use of antibiotics. And we continuously have to reduce not only the use of antibiotics as in animal feed, but also the therapeutic use. So the veterinary use of antibiotics is being decreased. And the first to stop using antibiotic growth promoters in the feed was the EU. That was already the case in 2006. But we see that today this is something that is taking place all over the world. And so that brings us to the question, of course, why were these antibiotics present in animal feed? For the simple reason, of course, that this gave on average, 10% improvement on performance. And how can antibiotics improve performance? Well, there have been many different attempts to get different sorts of explanations for that. But in the end, of course, we know that antibiotics, they kill bacteria, or at least they inhibit the growth of bacteria. So that is why people have taken a very keen interest in knowing more about the bacteria. And when we focus on the bacteria, we should go back to the origin of the bacteria. And when you look at the Darwinistic explanation of evolution, it is clear that bacteria are have evolved on this planet billions of years before the higher animals arrived, so billions of years before the pigs were there, and let alone before humans were there, because we as human beings are a very recent phenomenon in evolution, of course, and maybe we are a very temporary phenomenon actually. So looking at this, we should realize that the pigs and the higher and the vertebrates in general have evolved in a world that was already full of microbes, and that means that the bacteria and higher animals, they have gone through millions of years of co-evolution. And when keeping this in mind, it is, I think, easier to understand how things work in the intestine and the absorption of nutrients. We should realize in the first place that the bacteria are not all pathogens, even if we were taught, in the past that we should use antibiotics and disinfectants and trying to kill bacteria. 99% of the bacteria are not pathogens, are not causing any direct harm to the host. But even without that, even the so-called harmless bacteria, when you come to think of it, the bacterial cell is composed of the same building blocks like a pig cell or a human cell. It's composed of amino acids, nucleic acids, lipids, and even the trace elements like zinc and copper and magnesium and iron. They're very, very similar needs in the bacterial cell as in vertebrate cells. And that means, of course, that even harmless bacteria, we are competitors for the same nutrients because we all need the same building blocks of our cells. But on the other hand, of course, we should not consider bacteria as all dangerous, killing microbes that we should defend ourselves against. So it's a complicated and delicate interaction between the higher animal and the microbes that are present in the environment. And we see that already very early in evolution, even an earthworm has developed a specialized organ, a tube with an inlet and an outlet, which is designed to absorb the nutrients in this specialized organ, which is the intestinal tract. So the intestinal tract has been highly conserved through evolution. And it is well established now that whenever an animal or even a human being swallows, with each ml being swallowed, roughly on average about 1 million microbes are introduced into the intestinal tract and passing through the intestinal tract. So that is a given fact. There is continuous influx of microbes in the intestinal tract. And of course, we all know that in the stomach, the production of acid will kill a number of microbes, but acid is not a very good antibacterial substance. If one would choose an antibacterial substance, one would definitely prefer an alkaline substance to an acid substance. Acid substances are not very good at destroying microorganisms, and we will clearly see later on why that is the case. But anyway, the number of viable microbes in the stomach is reduced by 1,000-fold compared to what is swallowed. And this is still the case in the small intestine. And so 1,000 microbes per ml is a very low number. If you come to think of it, even good quality tap water contains 1,000 microbes per ml. So the number of microbes of active microbes in the small intestine normally is remarkably low. And then again, when the intestinal content arrives at the cecum and the colon, these numbers go really up extremely high. You should consider that, especially the distal colon, is like a bacterial soup. So taking this into account, we know that there's a lot of research being done on the interaction between the microbes and the host and on the composition of the microbial population in the intestinal tract. But very often people forget mentioning that all these studies are really focusing on the microbes that are present in the cecum and the colon. And the situation in the small intestine is completely different. And we will see in a minute why that is the case. So we should realize that the structure of the intestinal tract with a stomach and a small intestine and then a fermentation vessel, as we can call this, is again highly conserved in evolution. The chicken has a stomach with acid production, a small intestine for the absorption of the nutrients, a ceca for fermentation, and so has the pig, and so have most animal species. And of course, in the pig, we cannot talk about small intestine without first focusing a little bit on the stomach, because we all know that the stomach in the pig is a very delicate organ, and things often go wrong. And pigs, just like humans, are very susceptible to developing gastric ulcers. So what actually happens in the stomach is illustrated nicely, I think, by this graph, which we generated a number of years ago, looking at butyrate-producing microbes that are swallowed. And when you follow a butyrate-producing microbe through the intestinal tract, you can see that when it's in contact with acid, you know, the full line, which is the culturable bacteria, it drops to undetectable levels, and then it picks up again after a couple of hours. When there's no more contact with the acid of the stomach, the microbes can become metabolically active again. But the mere contact with acids very often does not kill the microbes, but the consequence of that is that they become metabolically inactive. Unculturable means metabolically inactive. Whereas if you just look at the viability, not at the culturability, then you see that the microbes are still there. They've not been killed. This butyrate-producing strain, which we are investigating in detail in the lab, was not killed by the acid, but it became metabolically inactive for a couple of hours. We call this the viable but non-culturable state. This is one of the elegant ways by which the intestinal tract is taking care of the microbes not coming into competition for the absorption of the nutrients, because this is the time needed for the passage of the small intestinal content and for the absorption of the nutrients. So the microbes cannot use the nutrients which are designed for the host during the passage through the small intestine. Whereas when they arrive in the cecum and colon, they can start multiplying again and they can consume whatever is there. But what is there for them, leftovers, are the undigestible fiber fraction. So this brings us to fiber, and we all know that fiber is very critical in pigs. And this was just a very simple study, which we did a number of years ago, where we simply looked at fiber content of a feed formula for pigs, and we compared, the grinding of the feed, fine or coarse, to a fine or coarse particle size. And what you can see here in this table is the score of the lesions of the ulcers in the stomach. So a lesion score of 5 means that there is dramatic ulceration in the stomach. And you can see that with low fiber content and fine particle size, you get quite serious lesions, gastric ulcer lesions. That's not new, of course, everybody knows that. And when you compare it with coarse particle size, you can see that almost half of the pigs had absolutely no lesions in the stomach at all. That has been established many years ago. But what we did on top of that is that we measured urease activity in the stomach. And urease activity is a parameter which is an indicator of Helicobacter infection. In humans, they use this urease breath test. So you blow in a machine and they measure the ammonia that is being exhaled. And that is also an indicator of urease activity in the stomach. So an indirect indicator of you being infected with, in the case of humans, Helicobacter pylori. And of course, we know that Helicobacter pylori in humans is associated with stomach ulcers. Is that the case in the pigs? Well, we see that there is a difference in positivity for urease test between the pigs that have severe gastric ulcers and the pigs that have less gastric ulcers. And so we have also a number of years ago started culturing this Helicobacter from pigs, and I think we're still quite unique in having a collection of Helicobacter bacteria from pigs, which we named Helicobacter suis. And it is a bacteria that is present in pigs all over the world, just like Helicobacter pylori is present in humans all over the world. And usually more than 60% of the pigs are infected. Very common. It is very common to see that 90 to 95% of sows and boars are infected with Helicobacter suis. And so the interesting thing is that this bacteria produces a urease, which is used for its detection, but it also produces a gamma-glutamyl transpeptidase. And this is just seems like, as a very technical detail, but it is essentially important, because what this enzyme does is it allows the bacteria to consume glutamine from the feed. So that's an amino acid, not glutamate, but glutamine. And glutamine is actually the nutrient that is used by the epithelial cells of the small intestine. So we should realize that the epithelial cells from the small intestine don't use glucose as their energy source, but they use an amino acid, namely glutamine from the feed, as their energy source. But of course, when Helicobacter suis is consuming the glutamine, there may be a shortage of glutamine for the epithelial cells in the small intestine. And then, of course, you have a problem with absorption of nutrients. So taking this one step further, we compared pigs that were carrying Helicobacter suis in their stomach to pigs that were not carrying Helicobacter suis in their stomach. And we looked at the entire gastric microbiota using these omics approaches, as you know them. And these omics approaches allow you to, without culturing, allow you to look at the composition of what bacteria are present in there. And you can clearly see from the colors here already that there is a difference in the entire microbiome of the stomach between pigs that carry Helicobacter suis and pigs that don't carry Helicobacter suis. But more importantly is when you look at the blue color, the pigs that carry Helicobacter suis have a lot of Escherichia coli in their stomach. That's important, of course, because we all know that a number of strains of Escherichia coli can cause diarrhea and can cause infection of the small intestine. If these pigs carry Escherichia coli in their stomach, they are at risk of developing E. coli enteritis, which is a serious issue in weaned piglets. Another one, which I think is interesting, is that there is a bacteria there which is called Fusobacterium, and we have done further research on that. This is a pathogen which is still not very well known in the pig, but it looks like it has an important consequence also for the intestinal health further down the intestinal tract and even for the colon. But the conclusion altogether is that when pigs are infected with Helicobacter suis, they carry a lot of other pathogens in their stomach, which can then start colonizing the small intestine. So, so taking this one step further again, we used the mouse model and we infected these mice with Helicobacter suis, and you can infect mice indeed, and they become carriers of Helicobacter suis. And then we looked in these mice that were infected compared to mice that were not infected. We looked at the in small intestinal leakage using a technique which I don't want to discuss in detail now, but it's a standardized technique. And it's clear here that the mice that were infected with Helicobacter suis had small intestinal leakage. You know, this is what this graph tells you. It means that the small intestine is not functioning well when mice were infected with Helicobacter in their stomach. So when Helicobacter suis in the stomach triggers leakage of the intestinal barrier of the small intestine. Now, this was just introducing the effect of gastric health on small intestinal health. And now we'll focus on the small intestine. But before going there, I want to remind you that, of course, what we have done in recent years is we have selected pigs for high performance. When you come to think of it, 25 years ago, a litter of pigs with 9 or 10 piglets was a nice litter. If the farmer today has a litter of 9 or 10 piglets, he's very, very disappointed. So we have selected for high performance, which means that you can only perform better when you take more feed. And more feed intake means that there is more stress on the intestinal tract, because the intestinal tract is not very different from what it was 25 years ago. So it's still the same structure, the same function. So there's more stress, and especially more oxidative stress on the intestinal tract, which means that there's more risk of what we call dysbiosis. This is particularly a problem in recently weaned piglets. So, so keeping this in mind, we focus now on the small intestine. So normally there's only 1,000 microbes per ml in the content of the small intestine. And that's a good thing, because the small intestine is where the absorption of nutrients takes place through this epithelium, the epithelium that covers the villus of the small intestine. And so the nutrients can immediately be taken up by the blood vessels and by the lymph vessels and pass into the body. So when, we all know about the gut barrier, but this is a remarkable barrier, because there's no strong, firm, barrier there. It's really a functional barrier, which is a very delicate structure, because it's a simple layer of a simple layer of a singular epithelial cells covering the host tissues. So when I told you already that these epithelial cells, they need glutamine for their energy supply. And that is important, because in this way, the glucose can all be saved for the other cells of the body. And what we also should realize is that the absorption of nutrient is not a simple diffusion process. It's actually an active process. And the yellow triangles, which I put here in this cartoon, they symbolize the receptors which are designed for the uptake of nutrients. So we should realize that for each and every nutrient which the animal needs, so for amino acids, for each and every trace element, for everything that is required as nutrient for the animal, there are receptors taking this up from the intestinal lumen, passing it into the intestinal epithelial cell, and then passing it on to the lymph vessels in the bloodstream. With this, we call the transcellular pathway. So uptake of nutrients is an active process that is mediated by receptors. And so, so this is the case for all the building blocks which we need in the body. And then on the other hand, of course, if there would be diffusion of potentially harmful small molecules, this, what we see here is a histological section of the small intestine. So this is a villus, and you can see that there's this, can you see this brown line, brownish line on the covering the villus? This is an immunohistochemical staining for another receptor, which is called the PGP. PGP receptor is actually, what this receptor is doing is any small potentially harmful molecule is being pumped back into the intestinal lumen. So the uptake of nutrients is an active process, and the pumping back of potentially harmful substances is also an active process. And so when taking this together, in an ideal world, you would think that this would be sufficient to have an optimal functioning of this small intestine, because the microbes are metabolically inactive, so they cannot compete. The nutrients can be taken up by receptor-mediated uptake process, and potentially harmful small toxic molecules are being pumped back into the lumen. But as you know, in biology, nothing is absolutely perfect, and so you will always have some microbes still being metabolically active in the small intestine and being a potential threat to the host. And therefore, the host needs additional protection mechanisms in the small intestine. And of course, when you do a scanning electron micrograph and you look at the intestinal surface from the lumen at a very high magnification, you cannot see the epithelial cells. You even cannot see the microvilli. What you see is the mucus layer covering these epithelial cells. And these mucins is the first thing that is facing the microbes when they pass through this small intestine. And so when we should realize that these mucins, they are actually highly glycosylated proteins, and these glycosylation, these sugar polymers, they bind a lot of water, and that creates a gel, which is actually the mucus. But we should realize that these glycosylations are like branched polysaccharides, and at each tip of the branch, there is one sugar which is called sialic acid. That's why these mucins are called sialylated. So they have sialic acid at their end of the branches, just like the leaves on the trees. And so when you're passing over a tropical rainforest, you don't see the soil simply because the leaves of the trees are masking the surface. And that is also the case in the intestine. The mucus is masking the surface because of the sialylation of the mucins. But when you take away this sialic acid by an enzyme which is called sialidase, which is produced by microbes that are pathogenic, that can cause damage to the small intestine, and you take away the sialic acid residues, then you can see the surface. When you take away the leaves, you can see the surface. And that's how the pathogens can start attacking the epithelial cells. So they produce a sialidase. And it's remarkable that microbes that cause damage in the intestine, most of them produce sialidase. That is the case for coccidiosis, for instance. It is the case for Escherichia coli. It produces sialidase. The pathogenic strains of Escherichia coli are at least. It is also the case for Salmonella. So these are all sialidase producing microorganisms, including Clostridium perfringens, which is also a pathogen of the small intestine in young piglets. And so when we focus like what Escherichia coli does, like the enteropathogenic strains of E. coli, abbreviated EPEC, when they can see the villous surface, when the leaves of the forests are taken away, the microbes can adhere to this villous surface, and that's what E. coli does. You see all these bacterial cells, they attach to the epithelium, they cover the epithelium, and they disturb the absorption of the nutrients. What we also should realize is that any stress will cause inflammation and will cause leakage of the intestinal barrier. And what does that mean? Well, it means that when you look at the cartoon of the epithelial cells, in between the epithelial cells, normally there's a seal closing the gap between the epithelial cells. when there is intestinal leakage, like is the case when the animal is under stress, then this seal is less tight, and there's a possibility of microbial components and microbes passing in between the epithelial cells from the lumen to the tissues of the animal. And there's also leakage of fluid from the host into the intestinal lumen, and that will result in diarrhea. So what you can see here is these reddish dots. And these reddish dots are actually expressed on the lateral surface of the epithelial cells. So whenever there is gut barrier leakage, when the tight junctions are leaking, there is possible contact with these red dots, and these are the toll-like receptors. I think you should remember this term. Toll-like receptors is a very important family of receptors, which when they are activated, they trigger inflammation. And so what we should focus on now is like what happens with bacteria when they pass through the intestine and when they start multiplying in the small intestine. How can the host defend its epithelial cells against these bacteria? And so what the host can see is the surface of the bacteria. So when we focus on the bacterial surface and we make a cartoon of the bacterial surface, that's how it looks. So this is the inside of the bacteria, which is the bacterial cytoplasm. It is surrounded by what is called an inner membrane. Then you have a very dense inert layer, protective layer, which is called the peptidoglycan layer. And in this peptidoglycan layer, there are lipoproteins anchored to it. And then in the gram-negative bacteria, you have another outer membrane, which contains the lipopolysaccharide. We have all heard about lipopolysaccharide or endotoxin, of course. Well, it is remarkable to see that the toll-like receptor, the red dots which I showed you a minute ago, they contain toll-like receptors, like for instance, toll-like receptor 4, which can bind to the lipopolysaccharide of the gram-negative bacteria. Whenever they recognize lipopolysaccharide from the gram-negative bacteria, it will activate a toll-like receptor 4, which will trigger inflammation. And so lipopolysaccharide recognized by the toll-like receptor 4 triggers inflammation. And inflammation is what is happening whenever there is epithelial damage. Bacteria can pass through, they can even get in the bloodstream, and that is called bacterial translocation, or when it's a pathogen, it's called septicemia. So this is what happens when there's damage to the epithelial cells. This needs to be avoided, and therefore the epithelial cells have special protective system and defense systems. And so what actually happens is that the epithelial cells, they secrete a number of defense molecules. Some of them are called defensins, which kill bacteria, but some of them are not very well known and still very, very important. And one of the compounds secreted by epithelial cells is called, it's an enzyme called alkaline phosphatase. And what alkaline phosphatase does is it can destroy lipopolysaccharide. That is fantastic because lipopolysaccharide is actually a very robust molecule which cannot easily be destroyed. There's very, very few enzymes that can destroy lipopolysaccharide, but the alkaline phosphatase produced by the small intestinal epithelial cell is destroying the lipopolysaccharide. And by doing this, part of the bacterial cell wall is destroyed. What happens next is that the epithelial cells also produce a molecule called peptidoglycan receptor protein. And this peptidoglycan receptor protein is actually drilling holes in the peptidoglycan layer. And by doing this, it kills the bacteria because it's destroying its protective layer. And so by doing this, what is left then is dead bacterial fragments, peptidoglycan fragments. Now there's still more enzymes produced by the epithelial cells in order to defend the epithelial surface. And one important enzyme is, we've all heard about it, is lysozyme. And what this lysozyme does is attacking again these peptidoglycan fragments that are still hanging around there. And so it actually breaks down the peptidoglycan by cutting in between the sequence in between the N-acetylmuramic acid and N-acetylglucosamine. So breaking down these peptidoglycan fragments into the smallest, the minimal bioactive component, which is actually the Miramil D Peptide, which is composed of muramic acid and 2 amino acids. Then when this means that the attacking bacteria were completely destroyed. So this is an important signal for the host. This is an important signal because when the bacteria have been completely destroyed, it means that everything is safe. It means that absorption of nutrients take place. So the host mucosa, the epithelial cells, have a sensor system for Miramil D Peptide. When there's a signal of Miramil D Peptide, the bacteria are destroyed, and it means that the epithelial cell can absorb nutrients, and there's no need for inflammation. And that is why Miramil D Peptide, MDP, Miramil D Peptide, it is sensed by a receptor inside the epithelial cell, which is called NOD2. And when that NOD2 is activated continuously in the intestine, there is reduction of inflammation, and this is what has been found in animal models and in human beings, that continuous long-term activation of NOD2 reduces inflammation and it has, as a consequence, better absorption of nutrients. And there's plenty of literature about this, but we don't need to go into detail there. So taking this together, the epithelial cells have sensor systems. They sense through the toll-like receptors when dangerous bacteria are penetrating in between the epithelial cells and may penetrate the bloodstream, and they respond to that with an inflammatory response. And inflammatory response also is associated with leaky gut. We should realize that this leakage of the gut barrier is already taking place simply when there is stress, as I told you, like, for instance, weaning stress. And that's why, recently weaned piglets are so susceptible to developing diarrhea. But it can happen before weaning, even when they're still under the sow. And so when this takes place, the villi become shorter and there is inflammation. Whereas when the situation is safe, it means that the epithelial cells are tolerant, they have strong tight junctions, they are numerous, and they develop long and slender villi, and there's very few inflammatory cells present. So that brings me to the conclusion and the take-home messages. We should realize that bacteria can compete with the host for the nutrients in the small intestine. Completely different from the cecum and colon. Small intestine is designed not for the bacteria, but for the absorption of the nutrients. The bacteria can pass through there inert, so without metabolic activity. They are flushed through with the intestinal flow, but they cannot come close to the epithelium because the epithelial cells are secreting these defense molecules, which I explained to you. So we have seen that the small intestinal epithelial cells have an elaborate defense system. So they're very essential for everything which happens in the small intestine. On the one hand, they are absorbing the nutrients through a receptor system. On the other hand, they have an elaborate defense system against bacterial colonization. And they are key in this antimicrobial defense. So don't forget, absorption of nutrients is going through the epithelium, and it must be fast and efficient in order to avoid loss of nutrients into the colon, because then nutrients are used to feed the undesirable population of microbes, and you get dysbiosis. So dysbiosis is actually a shift in the microbial population of the large intestine. Thank you very much. And I will hand the floor to Mathieu again.
Thank you very much, Richard. I think you made a very clear and simple explanation of a very complex subject. So thank you for that. It was very interesting. Before we move to the question, I just would like to remind to the audience that we have another webinar this afternoon. So you can see the title here. It's talking about nutrition and intestinal development in young piglets by Dr. Alphonse Jansman. And if you could move to the next slide. So this is the whole schedule of the different presentation. We will leave it there as we take the questions, so you can write down the time of the presentation you would like to attend. If you need the link, please ask us. We can send it to you. So now let's move to the question. So Richard, we have one question here, and okay, just would like to remind to everyone, you can write down your questions in the chat function. So we have one question which is actually similar to the question I wanted to ask you, so I'll ask only one question. So what would be the differences in microbiota composition between the small and the large intestine, and also during the life of the pig? How is it evolving?
Thank you, Mathieu. This is, I think, a very important question. As I explained in my presentation, the number of microbacterial cells in the small intestine should be very low. That is especially the case in the first part of the small intestine, which is the duodenum, and also in the second part, which is the jejunum. We see that in the ileum, so the lower part of the small intestine, the numbers go up. You know, there may be up to log 6 microbes in the ileum, but the microbes that are there are completely different from those in the cecum and the colon, and the ileum is separated from the cecum and the colon by the ileocecal sphincter. So there is a lock that separates the cecum and the colon from the ileum, and that's why the microbiota in the ileum is completely different from that in the cecum and the colon. In the ileum, the dominant population are lactobacilli and bifidobacteria, especially the lactobacilli. And what these lactobacilli and bifidobacteria do there is prepare the intestinal content for the feeding of the microbes in the cecum and colon. They break down a number of compounds into lactic acid. That's what lactobacilli do. That's what bifidobacteria do. We all know that. But what is the role of this lactic acid? It can be absorbed by the host, but that's not the important role. The important role of the lactic acid produced by the lactobacilli in the ileum is actually to feed butyrate producers. There's one family of butyrate producers which are called Lactospiraceae, and they convert lactic acid into butyric acid. So they live on the lactic acid produced by the lactobacilli and the bifidobacteria in the ileum. Whereas in the cecum and colon, lactobacilli should be a minority population. And then with respect to the evolution through the life of the animal, that is a very complex question. We should realize that in the pig, as in other mammals, as long as the dominant, the most important compound feed of the pig is the milk from the sow, then the dominant population in the cecum and colon is also lactobacilli and bifidobacteria. So we know that before weaning, this is just the major population, and it should be the major population. And so this changes quite rapidly as soon as the animal starts taking up hard food. And so this, the thing is that when you have an abrupt switch from a lactobacilli-dominated cecum and colon flora to a butyrate produce dominated cecum and colon flora, this is a difficult situation. And that's why we see so many issues with dysbiosis and diarrhea, scouring, when pigs are weaned. So we need to sort of prepare the piglets to this situation. And that is done through creep feeding, also preparing the piglets as they're still under the sow. But I think you will be, you will have much more experience with this, and you can explain to people more about this. The only thing I can say is that the effect on the microbiota of this is extremely important.
Thank you, Richard. So we've got quite a lot of questions coming out, so I'll try to take all of them. There is one practical question somebody was asking, how can they have the recording later? So it will be posted on Tonisity website, and if you have registered for the webinar, you will receive a link to access it. So, question you mentioned during your presentation about the importance of glutamine. So could you explain a bit more, please?
Yes, sure. So this is very, very remarkable, of course. Normally we know that most of the cells in the body, they use glucose as an energy source, or they use lipids as an energy source. The epithelial cells of the small intestine use glutamine as an energy source, although it is an amino acid, of course. And we know that, normally glutamine is plentiful in the feed. For instance, cereals contain, the proteins in the cereals contain a considerable amount of glutamine. So normally it's not, there's no shortage of glutamine, but it's still considered by nutritionists as a conditionally essential amino acid. That's what they call it, for the simple reason that when there's a shortage of glutamine, you will see that the performance of the animal will be poor, simply because the epithelial cells of the small intestine don't have enough energy. And so they will be fewer in number, and they will be less active, and they will be less active in absorption of the nutrients. And of course, this is worse when the pigs are infected with Helicobacter suis, because Helicobacter suis is consuming the glutamine from the feed. I can take it one step further by saying that glutamine is also produced by the animal itself in the stomach. That is because the glutamine is actually secreted by the acid-producing cell of the stomach. So normally the stomach is actually feeding the small intestinal epithelial cells. But when the Helicobacter is consuming the glutamine, well, then there may be a shortage of glutamine in the small intestine. So, so supporting the epithelial cells with of the small intestine with amino acids can be really meaningful and can be really useful.
Okay, thank you. Somebody is asking about the relationship between the presence of myeloperoxidase and enterotoxigenic E. coli.
Yes, that is an interesting question as well. So myeloperoxidase is actually an enzyme that is involved in defense against oxidative stress. So it is produced mainly by inflammatory cells, and this myeloperoxidase is actually inactivating oxygen radicals. I only touched on this briefly, but it is an important aspect. Inflammation is associated with oxidative stress. Inflammation is a defense mechanism against microbes. And so the immune system is killing the microbes, amongst others, by producing oxygen radicals. It's like bleach. Bleach is also a disinfectant that kills microbes, and it's also due to oxygen radicals. So it's really a very simple and broad spectrum defense system of the host producing this myeloperoxidase is actually defending the host against its own antibacterial substances. And so in case of E. coli colonizing the small intestine, the neutrophilic granulocytes, so the defense cells of the host, they are attacking the E. coli by producing oxygen radicals. And these oxygen radicals, they can also be harmful for the epithelial cells. So the epithelial cell is protecting itself by inactivating the oxygen radicals. And for that reason, of course, also other antioxidants like vitamin E and selenium and all that are also very important. So there is indeed a direct link between myeloperoxidase and Escherichia coli, because it's linked to the defense of the mucosa.
Okay, thank you. You mentioned at the end of your presentation that healthy piglets, they usually have longer villi, and let's say sick piglets which have diarrhea, they show shorter villi. Do you think that measuring the length of the villi is a good criteria to evaluate the function of the small intestine?
Well, there are different ways of evaluating the function of the small intestine, and these can be based on different analytical techniques, but usually you can only do it after the animal has been euthanized and necropsied. And that is also the case, of course, for measuring villus length. So that is the disadvantage, because you cannot do it on the live animal. But then when you look at different ways of evaluating the function of the small intestine, villus length measurements are indeed meaningful and are very useful, for the simple reason that the villus length is determined by the number of epithelial cells. And we should realize that the epithelial cells, they multiply in the crypts. So at the base of the villus, that's where the new epithelial cells are formed. And as the epithelial cell matures, it moves up the villus. And so the oldest epithelial cells, old, that's relative, of course, because they're only 2 or 3 days old, but the oldest epithelial cells are at the tip of the villus, and they're the ones that are most active in the absorption of nutrients. So they are most active in the production of antibacterial substances and most active in the absorption of nutrients. They're highly specialized. And so when the villus is shorter, there's loss of the epithelial cells at the tip of the villus. So it means that the most active and most valuable epithelial cells in the absorption of nutrients are lost. And that's the reason why there's a very good correlation between function of the small intestine, villus length, and performance of the animal, because it's really these epithelial cells at the tips of the villi that are most important in nutrient absorption.
Okay, thank you, very clear. I will take a last question. So somebody is asking if you have any comments on the intestine and lung axis relation in terms of immunity and overall health of the piglets when we are talking, for example, about viral infection.
Yes, yes. When we briefly focus on the viral infection, the most important viruses that attack the intestine, the pig, are rotavirus and coronavirus. TG, for instance, and of course, an epidemic diarrhea and related coronaviruses. They attack, and these viruses specifically attack the epithelial cells of the tips of the villi. So they don't even attack all the epithelial cells, only the epithelial cells of the tips of the villi. And so rotavirus as well as coronavirus do that, and even adenoviruses do that. But by attacking these epithelial cells, of course, they interfere with the absorption of the nutrients. And by doing this, attacking all the villi at the same time, the absorption of nutrients is so disturbed that all the nutrients arrive in the cecum and colon, and there they cause such a dramatic shift of the of the microbiota that you get a massive expansion of harmful gram-negative microbes like E. coli, Klebsiella, you name it. And that is actually the cause of the diarrhea scouring, which is due to rotavirus and coronavirus. So by attacking these epithelial cells at the tips of the villi, the villus will respond by contracting, and so that is why the villi become very, very short. But also by killing epithelial cells, they create gaps between these epithelial cells, which then allow activation of the toll-like receptors, as I explained, and that triggers inflammation and leakage. So you get fluid from the blood and from the lymph leaking into the intestinal lumen, and that's why piglets with diarrhea become so quickly dehydrated. You know, when you go in a farm and you have you know, rotavirus or coronavirus diarrhea, you see immediately that the eyes of the pigs, they are like retracted in and they are deep. So the eyeball seems to be retracted, and that's because the fluid behind the eyeball is lost because of the dehydration. It clearly shows you that the pigs die from dehydration. I hope this is answering the question, Mathieu.
I hope so, because I didn't ask it, so I'm not sure what the person was expecting. Anyway, thank you so much, Richard, for your time this morning. So for those who would like to attend the afternoon session with Dr. Jansman, it will be at 3 p.m. Central European time. Thank you again, Richard. It was a pleasure having you with us this morning, and we look forward to the next webinar. Thank you.
Thank you very much. Bye-bye.